All right, so I'll focus today on some basic concepts that have evolved over the past several years in terms of generation of hematopoietic stem cells and graftable hematopoietic stem cells from pluripotent stem cells. So in this classic view of the hematopoietic system, hematopoietic stem cells, HSC, reside, as you probably know, at the apex of this system. They have the multi-lineage capacity to produce cells of the erythroid, myeloid, or lymphoid cells. They also have self-renewal capacity, which places them in a good position to serve as a lifelong reservoir for whole blood stem cells. So given these unique properties, hematopoietic stem cell transplantation is evidently one of the most established cell therapy for hematologic disease as well as other disorders. Main sources of HSCs are peripheral blood, bone marrow, and umbilical cord blood. But there's a search for additional sources for HSCs, namely because of HLA match donors are not available for most patients in terms of allogeneic stem cell transplantation. In an autologous setting, a gene therapy setting, especially in the context of congenital bone marrow failure disorders, which is one of our main target disease, there's too few stem cells to actually genetically manipulate. And these stem cells are very difficult to expand using current protocols. So for these reasons, there is a clear need to develop and to produce engraftable HSCs from pluripotent stem cells, which would be of benefit not only for transplantation, but also for various other applications such as drug screening or even more basic biology questions, developmental biology, which are difficult to conduct, obviously, in a human setting for ethical reasons. So in vivo, the hematopoiesis develops sequentially in various locations. And in human, the blood starts forming around week 2 of development during gastrulation, a developmental phase that is defined by creation of layers, including the ectoderm, mesoderm, endoderm. Blood forms from the mesoderm. And because of a close developmental association between endothelial cells and endothelial cells, a hemangioblast has been proposed as a source or precursor for both cells. A cell that is or can be recognized by expression of KDR, as I'll get back to in a second. So these mesoderm migrate to an extra-embryonic region first in the yolk sac, defining the first wave of hematopoiesis, which is called primitive hematopoiesis. It is later on, around week 4 of development, that the first engraftable stem cells or stem cells capable of recapitulating or reproducing the whole hematopoietic system is observed in the AGM aortogonad mesonephros region of the aorta. These cells migrate to other sites eventually, but they expand in the liver and eventually establish residence in the bone marrow, where they establish long-term hematopoietic development postnatally. So these stem cells are thought to arise from hemogenic endothelium, or HE. HE are a subset of vascular cells that are found in the aorta, vascular wall of the aorta, dorsal aorta, and other sites as well. And through a process of endothelial to hematopoietic transition, these HE or these HSCs are specified and eventually bud out of into the subvascular region into the blood system. So the process of developing HSCs in vitro is mainly aiming at recapitulating these early phase or steps of the in vivo embryonic development from a pluripotent stem cells to a mesoderm, HE, and eventually hematopoietic stem and progenitor cells. So I'll use this little diagram over and over to go through some of the studies that have been conducted in that arena. So there are 2 general approaches that have been described to produce HSCs. The first one is called directed differentiation and consists in a stepwise addition of different cytokines and morphogens, such as those listed here, that will promote induction of pluripotent stem cells into a first mesodermal induction and then a hematoendothelial induction followed by hematopoietic stimulation or maturation at the end of the process. The second approach that has been described is called conversion, which also use a combination of cytokines and morphogen, but combines it with the overexpression of various transcription factors that have been shown to be involved in hematopoietic development. These transcription factors can be expressed at various stages of the differentiation process, either in primitive pluripotent stem cells or in derivatives produced during differentiation. A number of tissue culture processes have been described as listed here that most of you are probably familiar with, including the embryoid body system, all the way to an in vivo teratoma assay. The readout assays in our field are primarily based on demonstrating engraftment potential in an immune-deficient mouse and SG mice, but also immunophenotypically showing multilineage potential as well as T lymphoid production. A CFU assay at the bottom is defining a progenitor population, and a hemoglobin switching from embryonic and gamma globin to an adult globin is often used to define a definitive hematopoiesis. So starting with the first approach, the initial attempts at using the directed differentiation approach clearly led, and that was shown uniformly by multiple investigators, to a wave of primitive hematopoiesis with very limited engraftment and multilineage potential. And this led to the conclusion that definitive hematopoietic cells must emerge in order for these processes to be valid for production of Hscs, and also that we should be able to distinguish between primitive and definitive hematopoiesis. So these are some of the studies that have been published in an attempt to enhance hematopoietic or definitive hematopoiesis, and I'll review some of these along the way. So just for presentation purposes, we can group these studies into two groups: those that have attempted to recapitulate the microenvironment and those that have tried to modulate signaling pathways, even though some of these studies are overlapping. So all of these studies have shown a nice ability to produce definitive hematopoiesis. And I'd like to focus first on one of them. So which is one of the seminal studies that was published by the group of Gordon Keller. What they showed here is that they were able to, for the first time, distinguish and promote a definitive hematopoiesis and distinguish it from the primitive. And here's how they did that. They hypothesized that primitive and definitive hematopoiesis must be specified early in the differentiation process at the mesodermal stage of differentiation. And what they found is that when they inhibited the nodal pathway using a small molecule, SB as a nodal antagonist, and activating the Wnt beta-catenin pathway using a KIR agonist, they were able to promote definitive hematopoiesis as demonstrated by appearance of T lymphoid potential at the expense of the primitive hematopoiesis. They also found a nice approach to distinguish or to be able to physically separate the primitive from the definitive hematopoiesis by the use of a marker CD235, CD235A negative cells being faded to a more definitive hematopoiesis as we wish to achieve. Unfortunately, this was not sufficient to lead to production of engrafting hematopoietic stem cells, and same fate was also found for the other attempts at generating Hscs using this approach. In the first group, where people have attempted to recapitulate the microenvironment, there are some green check mark here suggesting that there has been some success. The first one, published quite some time ago in 2008, used an AGM stromal layer to produce engrafting stem cells. They showed some success, although this study has never been, to my knowledge, followed up on and included some large or high concentration of serum that might have been difficult to reproduce. I'd like to focus on two studies that were shown to produce some hematopoietic stem cells with some engraftment potential to some level. The first one came in 2013 in the form of teratoma, and this was published by two groups, and I'm showing one of the two here. So in this study, they injected subcutaneously or in the testicle of immune-deficient mice a combination of pluripotent stem cells, OP9, as well as microosmotic pumps to deliver hematopoietic cytokines during the teratoma formation process. 12 weeks later, they found hematopoietic cells that were similarly developed within the teratoma and that migrated to organs, hematopoietic organs in the mouse. And they showed this in this study at low levels. They were able to detect low levels human cell engraftment in the blood and marrow, and even showed some beta globin production. Importantly, when they took these cells from the teratoma and transplanted them in an immune-deficient mouse to assess for engraftment potential, they showed some relatively robust, up to 5% engraftment in these mice, suggesting that they had reached a true stem cells with multilineage potential. Another interesting study came from Gori and colleagues in 2015, where they use a two-step approach to generate engrafting stem cells. The first step involved a directed differentiation EB process where they generated or enriched for cells of the HE phenotype. These cells were sorted and plated, co-cultured on a vascular or endothelial cells that were also expressing surface JAG1 and DLL4, which are Notch ligand. So this was based on evidence that endothelial cells form vascular niches that are important for hematopoietic cell maturation, and that this process is also guided by Notch signaling. What they found is that when they took these cells 7 days later, they had a significant increase in hematopoietic yield compared to cultures with cytokines only, or even with cultures, co-cultures where the Notch ligand had been knocked down. They evidently showed some increase in Notch target genes, including shown here RONX1 and GATA2, which are well known to define definitive hematopoiesis. But more importantly, they showed that these cells, when transplanted in NSG mice, were capable of a sustained engraftment at levels of approximately 10%. Interestingly, they showed that these levels were similar to bona fide HSPC, a finding that, to my knowledge, has never been reproduced. And the levels of engraftment were significantly higher than when cells were cultured with cytokine only, and they demonstrated multilineage engraftment. So this study was published in 2015, to my knowledge, has not also received any follow-up studies, so this needs likely to be validated. So even though some evidence of engraftment was demonstrated in some of these studies, clearly none of these have clinical relevance. For example, the teratoma is definitely not an approach that could be used in patients. And for that reason and others, people have decided to use a second type of approach, which I referred to earlier, which is called the conversion strategy. And again, a number of investigators have tried to overexpress various transcription factors to help in the process of generating engrafting hematopoietic stem cells. Again, for presentation purposes, I grouped them into two groups: those studies that try to enrich or to overexpress single transcription factors, and those that try to overexpress multiple transcription factors. We can also look at them based on whether the transcription factors chosen were overexpressed directly in pluripotent stem cells or in derivatives of pluripotent stem cells, such as the HE or even hematopoietic progenitor cells that were then respecified to an HSC-like phenotype by overexpression of various combination of transcription factors. So I'd like to just briefly mention two seminal studies that were done in the early 2000s that really set the precedent for this approach. The first study tested or investigated the role of OXB4 overexpression in mouse, and that's the only mouse study I will show you. Embryonic stem cells. HOXB4 is a homeotic gene that gained some popularity in the field of hematopoietic stem cells in the mid-90s due to its strong ability to promote self-renewal. So the authors of this study overexpressed HOXB4 using an inducible system for only 2 days in HE, a cell derived from the pluripotent stem cells. And they did show significant levels of engraftment as shown on the bottom graph, left graph here. However, as you see on the right graphs, there was a marked myeloid skewing, and the lymphoid graph gradually waned off, suggesting that they had likely reprogrammed their cells or converted their cells to more of a myeloid type cell. But yet showed some evidence of engraftment. 3 years later, the group of McBadia at McMaster showed that when they tried to reproduce this procedure using human cells, a slightly different procedure, they considerably expressed HOXB4 at the HE level all the way. They found no evidence of engraftment in this situation. In fact, when they look at the molecular signature of Hscs derived from embryonic stem cells, they found that in fact the OXB cluster was overexpressed, and that in fact the OXA cluster was the one that was downregulated, especially the OXA 5 and 9, and I'll come back to this in a second. So there's been a large number of studies that have promoted or help in promoting hematopoiesis using various transcription factors. But as in the first case, very few actually showed evidence of engraftment. One of them, the SOX17, did not look and did not conduct transplantation studies. There's one interesting study that was recently published in PNAS where the transcription factor MLL-AF4 was overexpressed in progenitor cells derived from pluripotent stem cells. First, I was getting pretty excited when I saw the results. There was approximately 60% engraftment levels in the mice that were transplanted with these cells, and these were transplantable into secondary mice. Unfortunately, this was related to cellular transformation. Not a surprising finding, since MLL-AF4 is the product of the translocation 411, which has been associated with childhood lymphoma and leukemia. So definitely not a way to go. The only study using a single transcription factor that showed evidence of engraftment is one where the authors overexpressed RONX1A in pluripotent stem cells, and they found that approximately 10% engraftment at 9 weeks post-transplant, which is great, but there was also some suggestion, even though it's not clearly demonstrated in the manuscript, that this could also be due to a transformation, namely the majority of the cells that engrafted the mice retain an undifferentiated phenotype CD34, suggesting a block in differentiation consistent potentially with a leukemic transformation. So this led to the idea that possibly overexpressing multiple transcription factor would be beneficial to generate HSCs. The first study that is shown here is one by Dulatov and colleagues, where they identified a nice combination of 5 transcription factors that led to some interesting findings in terms of having cells with a short-term engraftment potential. But these cells were myeloid and erythroid skewed, and they showed that these were not true long-term repopulating cells. Same with Elcheva et al. In 2014, they identified these 2 combination transcription factors, but were not able to generate engrafting cells. Finally, just going back to these Hox genes that were found to be downregulated in the previous studies I showed, these when they were overexpressed in progenitor cells derived from pluripotent stem cells, unfortunately, were not sufficient to lead to engraftable cells either. So this leads us to the last study, which is the only, at the moment, the only study that shows evidence of engraftable stem cells convincingly without evidence of transformation. And I'll show you briefly what the authors did. to complete this. So they use a 4-step process where pluripotent stem cells were placed in a directed differentiation EB system to generate HE, hemogenic endothelium, and note the use of KIR and SB from the previous studies to promote a definitive hematopoiesis. These cells were sorted and found to be enriched in CD235A negative, which, as I showed before, are cells fated to a definitive hematopoiesis. They took these cells in a second step, which were considered to be able to induce a endothelial to hematopoietic transition in a period of 3 days, and took these cells and transduced them with a series of transcription factors, a library of 26 transcription factors that were cloned in an inducible lentiviral vector. These cells were immediately transplanted in NSG mice, taking advantage of the in vivo microenvironment to complete the maturation of the cells. Out of 40 mice that they transplanted, 5 were engrafted, as shown here, 1, 2, 3 in the first group. The other 2 mice, R and L, refers to the right and left femur where the cells were transplanted. So by using a PCR approach, they were able to identify a series of 7 transcription factors, as shown in the red box, that were common to all mice in all lineages. And using a factor -1 approach, they found that at least 5 of them were minimally important for induction of engrafting stem cells. So they repeated the process with only these 5 or 7 transcription factors, and these are some of the mice post-transplantation. Approximately 40% of their mice were engrafted, and some of them showed multi-lineage engraftment, and they were again able to bring these cells to a secondary transplant. So these results are, to this stage, the best in the field. However, there's still some limitation in terms of the cells that are derived from these, using this approach, are functionally distinct from, for example, cord blood, which is shown here in the inset there, which generates much higher levels of engraftment than these cells. They also show that these HSC-derived pluripotent stem cells are molecularly distinct and resemble more an HE phenotype, meaning that they may not have been fully converted to an HSC. The transformation potential is not addressed in this paper, but certainly something to consider. And also an important point is that the transcription factors that are seem to be specific to an EHT cell or cells undergoing EHT, if they transduce these transcription factors at the HE level prior to inducing endothelial to hematopoietic transition, they lose all the engraftment potential. So this is where we're at the moment in the field, and obviously there is some very interesting findings and promising studies, but I would judge that none of these really have significant clinical applications and that more work needs to be done. So what do we should keep in our notebook from this? Hopefully not this. We found that there was some interesting, promising studies using both a directed differentiation approach as well as a conversion approach. The field seems to be moving in the use of multiple transcription factors to generate engraftable stem cells. Some important points to show: it's usually said that it is difficult to recapitulate the in vivo process. This is why it's difficult to generate engraftable stem cells, which is, I think, true, and certainly we should encourage studies to promote knowledge of developmental biology. But I would just point out one study that has been deposited in bioRxiv some time ago from a French group that proposed an approach, a single-step approach, where cytokines only are used for the differentiation with seemingly no attempt at recapitulating the different stages of embryonic development, and they propose some pretty good results, but we'll see if this, once it's published, what the story is exactly. And the other points are self-evident, but one of the main things is that it looks like a pluripotent stem cells derivative seems to be a cell of interest, such as the HE, probably because these cells minimize the epigenetic distance while maintaining the scalability of pluripotent stem cells, which we lose by using somatic cells. The road to clinic is probably a little far still. A lot of work probably to improve efficiency, to improve safety and others. But in terms of efficiency, I think an understanding of cell identity, of gene regulatory network cell identity would be of value. And with the identification of super enhancer, we have a way to do this now. Likely we'll need to introduce some epigenetic modulators to allow or increase heterochromatin access to various transcription factors, unless we can find pioneer transcription factors that can bind directly to heterochromatin and allow access to all transcription factors that would be necessary for HSC production. So I'll, and then there's other topic that will need to be addressed as well, which I don't have time to go through. I just would like to finish with this slide if promoting our research postdoctoral fellowship. Anybody interested is welcome to apply, come and see me or email me. Thank you very much for your attention.
Please come to the microphone if you have any questions. I'll start off with the first one. So the model systems for evaluating these, like the NSG model, are really not faithfully representing the microenvironment that these cells need. What do you think the best way to improve the models would be in the short term?
Right. Yeah, so they're definitely not ideal models, although we know as a first point that the somatic stem cells do engraft and do produce multilineage engraftment. So in that sense, it's a model that we're trying to reproduce. In terms of better models, certainly moving to a large animal model where an autologous transplantation can be done would be likely of interest to really demonstrate long-term engraftment potential for these cells. Yeah.
Sorry, actually I have a follow-up. Bless just ask a question I want to ask, so I have a follow-up. So if you're talking about if we should also try the non-human primate, like large animal model, what would be the like clinical relevant number of HSCs that we have to achieve? And the current differentiation method or using bioreactor, do you think it's already getting the number besides engraftable?
Yeah, likely with the current approaches, we're pretty far from the clinical scalability that we'd need. Some have estimated to about 1,000-fold increase in productivity that would be required. So we're, I think, pretty far from this. In terms of number of cells that would be needed, I think we usually think in terms of CD34 positive cells. In terms of we usually transplant, at least for primary cells, approximately 5 million per kilogram. So you can make the math and try to see how much work we still need to do to scale up the process. Yeah.
Any other questions? Thank you very much.